Hi, my name is Dr. Adam Nathan McShane from the Wellcome Wolfson Institute for Experimental Medicine, Queen's University, Belfast. I'm going to describe our paper entitled SorLA Regulates B-cell receptor uptake, trafficking, B-cell mediated immune responses in vivo. A high-affinity antibody response to both infection and vaccination critically relies on the ability of B cells to capture and process antigen for presentation to CD4 plus T cells. However, the molecular composition of B-cell receptor-antigen endocytic and intracellular trafficking machinery remains poorly understood. Here we characterize the role of intracellular trafficking receptor SorLA as the novel regulator of B-cell receptor-antigen trafficking. We find that SorLA localizes to both the cell surface and endolysosomal compartments, and increasingly localizes to compartments associated with antigen processing following antigen uptake. We also establish an interaction between SorLA and the B-cell receptor in both Ramos cells and primary murine B cells. To investigate SorLA's specific role in B-cell antigen uptake and trafficking, we deleted SorL1, which encodes SorLA, in Ramos-Castan B cells, and analyzed primary B cells from SorL1 knockout mice. SorLA deletion resulted in increased surface B-cell receptor levels, reduced soluble and surface-bound antigen uptake, downstream antigen trafficking defect through compartments associated with antigen processing, and reduced B-cell receptor antigen degradation. This resulted in reduced delivery of antigen to compartments associated with MHC class II loading, and using SorL1 knockout Ramos-Castan cells expressing B-cell receptors with specificities of HIV-1 broadly neutralizing antibodies, lower levels of HIV-1 envelope-derived peptide presentation. To investigate how this influences B-cell mediated immune responses in vivo, we immunized SorL1 knockout mice with a T-dependent antigen. Lack of SorLA led to reduced antigen-specific germinal center B-cell numbers and plasma cell frequencies, and lower levels of antigen-specific IgG1 antibodies, delayed affinity maturation. Here we present the first account of the critical role of SorLA in immunity. Together, our results provide evidence of a novel role for SorLA in B-cell receptor antigen trafficking and the modulation of immune responses in vivo. Thank you for listening.